We'll give it another minute.
Hey, you like the image? You taught us how to do it.
Oh,
You may not remember, but we got the protocols from you.
Is this our platform?
Yeah.
Oh, it's
We use it
beautiful.
all the time now.
Yeah.
We don't like 32 markers. It's a little too many for us, but 12, no problem.
Yeah, it's beautiful. Well done. Okay, it's 8 o'clock. Thank you everyone for joining us as we begin a new academic year for the Myeloid Network. On behalf of Judy Varner, Dmitry Gabrilovich, Jennifer Guerrero, and Joanna Joyce, it's our pleasure to welcome you to the series. And we have the pleasure of having Dr. David DeNardo today lead us off for this new season. Dr. DeNardo did his PhD at Baylor College of Medicine in cell biology, studying nuclear hormone receptor signaling. And then he joined my lab as a postdoc fellow when we were at UCSF and made some seminal observations regarding how myeloid cells are programmed in tumor microenvironments, recognizing that it's IL-4 and IL-13 from CD4 helper T cells that provide the Th2 type programming to macrophages that engage their pro-tumorigenic activity. He then went on to recognize that by targeting macrophages and one of their key survival pathways through CSF1 and CSF1 receptor signaling, that inhibition relieved the T cell suppression and engaged a productive and cytotoxic T cell response to turn tumor progression and tumor kinetics down significantly. At that point, David started his own lab where he has just done a phenomenal job with evaluating myeloid biology, mostly in the context of pancreatic carcinogenesis, where his lab is at Washington University School of Medicine in St. Louis, Missouri in the United States. He's a member of the Siteman Cancer Center. And in that context, he's a program leader for the Cancer Center's Tumor Immunology program. His laboratory over the last 10 years has focused on three major areas: understanding the role of fibrosis in controlling, contributing, and regulating tumor immunity in predominantly the pancreas; therapeutically reprogramming myeloid and dendritic cell responses to improve responses to standard of care and other immune therapy targets; and then also really just in a global way, trying to understand the impact of myeloid cell heterogeneity on tumor progression. Most of his models are in pancreas, but he does do studies in lung and breast. And if anyone is looking for a good postdoc mentor or just phenomenal graduate school mentoring, I highly encourage you to seek out David and look at the next part of your career in Dr. DeNardo's laboratory. So David, please take it away. The stage is yours. Thank you for talking today.
Thank you very much for the kind introduction and all of you for the invitation to present, and also for the plug for my lab. We are looking for postdocs. So I study the tumor microenvironment, as Lisa highlighted, and a large portion of what we do is in pancreas, so it's not the only model. Make sure I can run my slides. There we go. So this is not pancreas cancer. This is an H&E of melanoma, and I show it to remind us that there are a diversity of tumor microenvironments in the tumors we study. And when I look at this, this is a cutaneous lesion. Hopefully you can see my laser underneath the skin, and we can see tumor cells stacked on tumor cell with some modest but limited stroma. Pancreas cancer, by contrast, if you aren't familiar with the disease, is almost completely the opposite. These are the nest of tumors, and they're embedded in a desmoplastic response. That's an expansion of the otherwise genetically normal stromal cells surrounding this tumor. And in fact, 85% of the cellularity or cellular area within pancreas cancer is the stroma rather than the malignant cells. So when we're thinking about myeloid biology, immune responses, responses to treatment, or even immunotherapy in this disease, we have to think about this context and this organ-specific pathobiology of this disease, and it may change how these myeloid cells in this context of today's talk respond and what they do. In addition to being highly desmoplastic, it's highly fibrotic with dense extracellular matrix. I like this staining for trichrome. It's a collagen staining of blue. You can readily see that there's abundance of collagen in these tumors. They're often described as very solid by the surgeons when they excise them in our group, and you know, they're more like scar tissue that grows. But they're not without immune cells. Here, a pan-myeloid marker, CD68, the same image I showed you the H&E from. Myeloid cells are often abundant in these tumors, as well as other CD45 components. And while this tumor is notoriously unresponsive to various lines of immunotherapy, including checkpoint, about a third of these tumors have significant T-cell infiltrations. I just show you our favorite picture of this. And they correlate with good clinical outcomes. What we don't understand is why we're unable to leverage this for at least benefit for a subset of these patients. So some of what I've said in the past, multiple times, has suggested this fibrotic tumor microenvironment in which these innate immune cells, including myeloid cells like macrophages or dendritic cells, are embedded, might give them unique tissue remodeling and anti-adaptive immune type properties. We've had this hypothesis in the lab for a while and studied this in mechanistic details in various studies, but we've been unable to appreciate how this would look compared to other cancers. And this is a unique feature of pancreas cancer. So fortunately, why is that running? Very good. Fortunately, I've had a very talented physician scientist in the group who really focuses on genomic and analysis. And he's taken advantage of the fact that now beyond our institution's own data sets, that you can curate data sets for single cell analysis across a myriad of human cancers. And in fact, he built a human cancer macrophage atlas from, I think, about 15 studies, including 70 tumors from 11 different tumor types, including 54,000 tumor-associated macrophages, and a large group of pancreas tumors from our own institution. The first thing you learn is when you color code all these things, you learn the thing that my kindergarten girl has learned is if you mix all the colors, the main color you see in a UMAP is brown. But the more importantly, the thing we learned was how these different macrophages, based on the tumor microenvironments they might be specific to these disease types, might be different. We've done this in a couple different ways, but this is a simple GSEA analysis across these 54,000 macrophages, then striated or clustered by tumor type. What we can see, even looking at the hallmark pathways, is our thoughts on the tumor microenvironment that macrophages are in pancreas are not entirely wrong. We see high levels of elements that would be for macrophages in a fibrotic environment, such as Wnt or sonic hedgehog signaling, high levels of glycolysis and hypoxia, which would go together, and chronic activation of pathways like NF-kappa B. And relatively to other cancers, relatively poor responses to ROS, oxidative phosphorylation, and interferon. This does suggest that fibrotic tumor microenvironment is actually uniquely poisoning macrophages with certain phenotypes within pancreas cancer that, while not entirely distinct from other microenvironments, but at least maybe on whole at one end of the spectrum compared to other major cancer types. And we've analyzed this in a couple different ways. So with this as a backdrop and these two sides, fibrosis driving macrophage phenotype or fibrosis interacting macrophage phenotype, and a lack of sort of ROS interferon signaling, I thought I'd talk about two as of yet unpublished stories from the laboratory. One of them more basic science, and one of them clearly translational biomarker driven. First story is around origin and architecture. So before I get into origin and architecture, if you're not a pancreas person, you need to understand a few pieces of the anatomy of the pancreas that I had to learn when I went from breast to pancreas in terms of a learning program. This organ, which often attaches to the spleen in the mouse, actually one of my colleagues considers it the booger that attaches to the spleen. It's actually really important for two biologic functions. One is insulin to sustain blood sugar. This is done by the islets within the pancreas. And the other is for secreted enzymes that help you digest some of the things in your digestive system that go into the digestive tract. These are the acinar cells that secrete these sort of granules of digestive enzymes that then are shunted into your digestive system. And both of these functions are absolutely critical. Now, surrounding these acinar cells, which will be the focus of today's talk, are two different cell types that are really critical for their biologic function on a day-to-day basis, but also for their maintenance and homeostasis. This includes a fibroblast subset, which are often termed pancreatic stellate cells, as well as tissue-resident macrophages I'll show you in a minute. And they're important for the homeostasis of these acinar cell biology. Now, when pathology happens in the pancreas, whether this is in the context of cancer, such as pancreas cancer, or with tissue injury due to a myriad of insults, either alcoholic or chemical, or even a chronic inflammatory in the context of pancreatitis, but in the absence of cancer, there are two things that happen. This tissue becomes highly inflammatory. This is human tissues. You see large numbers of macrophages expanded. You see a highly fibrotic microenvironment, as I showed you in the cancer, but also true in pancreatitis. And there's an expansion of fibroblasts. So this expansion of macrophages and fibroblasts and the extracellular matrix happens all in parallel during tissue damage and loss of homeostasis in cancer and non-cancer related inflammatory conditions in the pancreas. This also can be mimicked in the mouse models. Here we're showing in experimental pancreatitis and an enhancement of both fibroblasts as well as tumor-associated macrophages. Cancer models look the same. Now, I think this is a very old slide at this point. And many of you already, all of you probably already know this. Macrophages come from diverse origins and can be seeded into tissues, at least in animals, in these ways. This includes what we've always thought about, development from hematopoietic stem cells in the bone marrow predominantly, where blood monocytes can traffic into tissue and become tissue resident to those tissues. But also early on in development, embryonic progenitors can lead to a subset of tissue resident macrophages that are seeded into here an agnostic tissue. Now, this varies widely in terms of which of these subsets contributes and how long they will contribute for during the duration of the animal's lifespan. Such as in the colon, it's predominantly HSC, the turnover of macrophages is much more high, and they're replaced rapidly. Versus in the brain, by the under contrast, microglia are thought to be seeded by embryonic origins, and then at least retained for a long period of time in the absence of inflammation in that setting. Other organs such as pancreas, liver, lung have more intermediates where subsets of these macrophages can come from either origin. Under stress, you can have expansion of macrophages through two of these mechanisms at least. You can have a recruitment of monocytes, as many people have shown, into the microenvironment, whether this be an inflammatory environment or whether this be a cancer. And you can have expansion of otherwise tissue-resident macrophages, no matter whether they're embryonic or HSC in origin. And both of these occur. Now, a number of years ago, we showed in the context of a pancreas tumor model that there's an expansion of embryonic-derived progenitors, embryonic-derived macrophages. They expand in the tumor microenvironment, and they actually have distinct phenotypes around shaping the fibrotic response. However, John Baer, this was primarily in transplantable models, and John Baer wasn't fully satisfied with the details of the study, which was sort of a first report a number of years ago. And so he wanted to follow up on that. He did this partly because the biology of tumorigenesis doesn't occur in isolation by a grafting mechanism that we often do in animals, put in the tumor cells, watch them grow. But rather, there's an acinar ductal metaplasia that often is driven by the inflammation or the induction of KRAS oncogene. Then inflammatory processes along with the oncogenes collaborate to lead to progression and a full ductal phenotype in the eventual cancer. And this is often spurred or accelerated by the elements of pancreatitis or chronic inflammatory conditions that happen in the pancreas, and paralleled by augmentation of fibrosis. So these elements might all work together. Therefore, origin-based and macrophage subset-based studies should happen in this context. At least that's how John Baer said it. There's another feature that is quite remarkable about the pancreas. So we can experimentally induce pancreatitis. We do this with cereulin, which blocks the secretion of these acinar cells, and actually they undergo morphogenesis, cellular stress, undergo ductal morphogenesis, and eventually die if you sustain it for long enough. This absolutely shreds the pancreas. You'll lose about 40 to 50% of the pancreas area in some of our models. And you can see this by looking at the H&E. The acinar cells also undergo acinar to ductal metaplasia or morphogenesis transition. But what is remarkable, if you leave the fibrosis and inflammation happens at the same time. But if you leave the pancreas another 7 days in the absence of the stimuli that's causing the pancreatitis, this organ is able to completely heal itself. So the H&Es from the pancreas after 10 days of recovery are indistinguishable from the H&Es from an untreated mouse. So this suggests that in this organ, we have a very powerful wound healing and homeostasis program that's able to go from the edge of collapse and back to complete homeostasis over short periods of time. And we believe this is through the co-regulation of macrophages and fibroblasts. I'm going to show you some of that data today. So with this background, John first, because we hadn't done this in non-cancerous pancreatitis models or in pancreatitis accelerated cancer models, he first looked at several lineage tracing models to understand where macrophages come from in this context, including 3 different lineage tracing models. I'm going to use FLT3 Cree-driven model here just as an example throughout the story, but he's done it in several different models. I'll occasionally show you other ones. So for the FLT3 Cree-driven models, this allows you to identify HSC-derived myeloid cells, and actually all CD45 cells, as they go through a FLT3 Cree positive progenitor. So any macrophages present in a tissue that are FLT3 Cree or YFP positive in this context are derived from predominantly HSC mediated hematopoiesis and predominantly often monocytes. We think monocytes. By contrast, if they don't go through a FLT3 positive, this could be derived from an embryonic progenitor, although emergency hematopoiesis can also contribute. So where we have YFP negative, these are often embryonic macrophages. So if we look at microglia, they're predominantly YFP negative by contrast. So he did this in the context of our pancreatitis model. And what he found is under normal homeostatic conditions, there's about 30 to 40% of the macrophages within the pancreas are actually FLT3 negative or YFP negative, suggesting their embryonic origin. And this expands dramatically during cereulin, up to about 40 to 50%. But numerically, both embryonically derived and non-embryonic macrophages expand. But the embryonically derived or FLT3 independent macrophages expand most dramatically. They have about a 15-fold expansion in about 7 days. And this happens through absolutely high levels of proliferation, as seen by Ki-67, that are most high in the embryonically derived macrophages in the context of pancreatitis. So tissue-resident macrophages could be of embryonic, non-embryonic origins. The embryonic origin macrophages are expanding dramatically during pancreatic cereulin-induced pancreatitis. He used another model that was actually quite interesting. He pulse chased using the CSF1 ERT2 or mercury and a LSL TD tomato. This allowed him to look at total tissue resident macrophages independent of their etiology embryonic origin. He did this for 4 days, and then he chased for, I think this is about 70 days before then looking at the tissues. And under homeostatic conditions, he can then find that in this context, he can label subsets of tissue resident macrophages shown by the TD tomato staining. And then after this, if we induce cereulin-induced pancreatitis, there's a dramatic expansion of these tissue resident macrophages as shown here. But almost equally interesting is their location. We have subsets of these macrophages intimately involved with the acinar cells and subsets of these macrophages in these acinar interstitial spaces where fibrosis begins to appear in this animal model. And I think this will become important in a minute. So in total, these data suggest that both embryonic and total tissue resident macrophages expand during tissue injury in the pancreas. This includes pancreas cancer, as we published before, but also pancreatitis or pancreatitis accelerated pancreas cancer in data I'm not showing you. So then he went to phenotype these. We've done this now with a couple of models, but the first model we did was the FLT3 Cree-driven model. And we looked for the embryonically derived or non-embryonically derived macrophages. He could see significant transcriptional differences, whether it be normal pancreas, pancreatitis, or in pancreas cancer transplantable models. And when he looked for the GO terms or GSCA terms of this, it's recapitulated what we had published earlier, is that there's a significant amount of genes involved in extracellular matrix production, interaction, integrin signaling, upregulated, and those that we would predict to be more tissue resident and embryonically derived, versus the YFP HSC derived are more likely to have genes involved in antigen presentation or immune stimulation. But bulk RNA-seq can lie sometimes or obscure the truth. So we redid this by single cell RNA-seq analysis, and this ends up being critically important for the rest of the story. First, I'm just going to show you a positive control. So you do this in a liver. This happens to be a PDAC-seeded metastatic liver, and we have the YFP negative tissue resident macrophages. We get distinct UMAP clustering of these cells, and all of these 2, 0, 3. This ends up being the liver Kupffer cells predominantly. and only a few other clusters appear. But if you look at the YFP positive subset, the HSC derived, you end up with macrophages that are distinct from these YFP negatives, and these end up being not the Kupffer cells, and then there's one shared origin cluster. So in organs like that we've looked at, like liver and lung, we see that the origin succinct macrophages are preserved even in the cancer setting, and that transcriptionally they're distinct, at least over these time periods. The pancreas, this is not what we saw. What we find is that there are various different transcriptional clusters shown in the UMAP, and that these clusters intermix highly between the two different origins as sorted by the YFP lineage tracing system. So as the embryonic macrophages are present in every single cluster, transcriptionally we see, suggesting that the overall clustering phenotypes or subsets of macrophages are not purely seeded by their origin, but by probably their tissue-specific contexture and their microscopic localization. Now, there is some subtlety in this. If we look at this as cluster 0 ends up being our main tissue-resident macrophage cluster, there are still subtle transcriptional phenotypic differences as shown by the mild movement in the UMAP that we can see in GSCA that focus around still extracellular matrix interaction and different genes like this. There are only 1 or 2 subsets that are actually uniquely distinct. This includes this cluster 2, which is predominantly maybe 85% derived from the embryonic origin. This ends up being a cluster of macrophages highly marked by live-1 and probably responsible for much of the transcriptional patterning we see in bulk sequencing. Because when we then compare cluster 2 to all the rest of the macrophages, independent of their tissue-resident or IP positive or negative, what we find is that it's highly enriched for the extracellular matrix interacting and extracellular matrix remodeling or tissue remodeling type phenotypes, and lower in antigen presentation immune interacting. I show you this in pancreatitis, but very true also in normal pancreas and pancreas cancer single cell subsets with some distinction. So where are they located? So what I told you before is when we label total tissue resident macrophages, just check the time, okay. Total tissue resident macrophages, we find them both intimately interacting with the acinar cells and also in these interstitial spaces where fibrosis tends to begin. When we look for live-1 macrophages, either in multiplex or simply shown here by a single live-1 stain, we find these macrophages are actually only in the interacinar space and not intimately interacting with the acinar cells themselves. Before I show you maybe a model and characterize that a little bit further, mice can lie, so we always have to check in humans. So we've taken advantage of single cell data sets for human chronic pancreatitis, as well as our own internal data sets for human pancreas cancer. We've mapped both the live-1 gene and live-1 gene signature from mouse into these data sets, and we find that there's a unique cluster of live-1 positive tumor-associated macrophages in pancreatitis or tumor-associated macrophages in cancer. And these clusters are fairly unique within these contexts. When we then look for either marker genes or for the GSCA terms for their phenotypic changes, we see that it's very similar to the mouse. Some of the same marker genes come out as distinct in these clusters. They're live-1 marking, and some of the same extracellular matrix and tissue remodeling programs are highly upregulated in these live-1 macrophages in pancreatitis or pancreas cancer in human, and are very similar to those programs we saw in the mouse model. So the mouse was not entirely wrong in this context. The localization also is very similar. We try to co-mark, in this case, macrophages with one marker here, one CZ163, and live-1. We find that the predominant live-1 positive macrophages are present in these acinar interstitial spaces in pancreatitis and not present in intimate interactions with the acinar. Reminding you that when we look at fibrosis like extracellular matrix or CAFs, they're present in this acinar interstitial spaces, or these in-between acinar cluster spaces. Similar phenotype was also seen by IHC in pancreas cancer. I won't detail that too much. So together, these data, transcriptional and phenotypic and lineage tracing, suggests that, tissue-resident macrophages absolutely in the pancreas come from multiple origins, HSC and non-HSC. They expand dramatically during tissue injury, including pancreas cancer and pancreatitis. The live-1 subset is more embryonic in origin, at least in the animal model, suggesting at least a long-lived tissue-resident macrophage phenotype that would be retained in human models, and suggests they have high interaction with extracellular matrix and fibroblasts. They're located uniquely in these interacinar stromal, which would allow them to modulate the extracellular matrix or respond to the extracellular matrix, as well as fibroblasts. And other tissue-resident macrophages, as well as monocyte-derived macrophages, often are localized to these acinar clusters and intimate interactions, which probably has a distinct phenotypic or functional purpose. So how can we assign function, and what have we done there? I've taken two different models. I'll show you both of them today. One is to do a CSF1 clonidinate to combined depletion pulse chase to deplete total tissue resident macrophages and allow monocytes to recover. When we do this, we find that after a chase of about 10 days, we recover inflammatory monocytes in circulation, suggesting they're still there to be recruited to whatever inflammatory insult we give. But the tissue resident macrophages are down about 85, 90% within the pancreas. And then when we induce serolin-induced pancreatitis, we find there's an expansion of macrophages. This expansion is absolutely blunted when we've already taken out tissue-resident macrophages. So this model will allow us to assign functional impact of total tissue-resident macrophages. In a minute, I'll show you a live one specific model, but I'll show you the data from this. First thing we looked at, it's actually not the first thing we looked at, but the thing we looked at that will be first in this talk is the expansion of fibroblasts. So during pancreatitis, we see a dramatic expansion of fibroblasts, either by IHC as shown here, or by flow cytometry as shown on the bottom. What we find is when we remove tissue-resident macrophages by CSF1 clonidinate in this pulse chase format, we find that these pancreatic stellate cells are unable to expand into fibroblasts in the context of pancreatitis without tissue-resident macrophages, or impaired in their expansion across multiple fibroblast subsets. Now, there's some caveats to CSF1 clonidinate system. So we also employed more specific, a live one Cree-driven bred to a CSF1 flox mouse. So whenever live one is turned on, CSF1 is turned off, and this impairs the survival of live one positive macrophages within tissues. I'm not showing you here, this does deplete live one positive macrophages, but we repeated the experiment giving an injury model here, serolin-induced pancreatitis, and found that again, we have an impairment of the expansion of fibrosis as measured by pancreatic stellate cell activation by photoplanin here, as well as flow cytometry for total fibroblast numbers, which failed to increase in this model in the absence, or increase as much in this model in the absence of live one positive tissue-resident macrophages. Then finally, because there's actually some distinction in the IHC in terms of the total levels of the IHC markers we typically use for activated fibroblasts, such as photoplanin or PDGF receptor. They're much higher in terms of total protein level in the Cree-negative mice or in the control mice, suggesting there may be phenotypic switches as well at play. We did single cell analysis of the fibroblasts present within the pancreas, plus and minus tissue-resident macrophages using the CSF1 clonidinate system. When we do this, we can, of course, subset several different fibroblast subsets, which are probably immaterial for today's talk. Most abundant is this inflammatory fibroblast subset present within serolin-induced pancreatitis. But they're transcriptionally distinct, whether there are tissue-resident macrophages present, as in the control, or depleted by CSF1 clonidinate. When we look at those distinctions, is that in the absence of tissue-resident macrophages, we see an absolute loss of their ability to produce extracellular matrix and extracellular matrix reorganizing molecules, and a significant augmentation across all fibroblast subsets of production of interferon and stress-related inflammatory pathways, suggesting that these fibroblasts are numerically impaired, but also phenotypically no longer able to do their tissue remodeling job and preserve homeostasis, at least in theory. So this is our working model. Both tissue-resident macrophages probably play a role here. Live one positive tissue-resident macrophages are able to expand these pancreatic stellate cells to allow them to probably protect and preserve tissue homeostasis. But I haven't showed you the tissue homeostasis part yet, so let's do that. To do that, John used a pretty severe model of pancreatitis. This is to put an osmotic pump with our serolin-induced pancreatitis moiety or a molecule. When he does this, even in control mice dramatically lose weight to about 20%, which is our endpoint for this type of study. But then, as I suggested, even as the serolin wanes, they are able to regain their weight, not always up to full potential, and they survive this nearly 100% of the time. So in spite of a severe pancreatitis, they're able to restore homeostasis to the pancreas. I showed you the H&E earlier. They actually look quite normal pancreas at the end of this. By contrast, if we deplete total tissue resident macrophages, we find that they're absolutely unable to restore homeostasis. They progressively lose weight until they're non-viable and we have to sack them. As an endpoint, this is their survival. So if we lose tissue resident macrophages, they no longer can survive cereulin-induced pancreatitis. And then we've checked by autopsy in a couple different methods to make sure this is a pancreatitis specific event, and it appears to be, and has the same kinetics as if we when we ablate the pancreas as a whole surgically. I'll also just mention this is in contrast to other sort of macrophage or monocyte depleting methods. This CCR2 knocks out a majority of inflammatory monocytes in circulation, about 85% of the way, although they still can reconstitute and circulate this last remaining percentage. These mice actually survive this pancreatitis model quite well, suggesting that monocyte-derived macrophages have a lesser degree or lesser role in this tissue homeostasis program within experimental pancreatitis. It's just showing you some of the data that the pancreas, in the absence of tissue-resident macrophages, are absolutely liquefied. Acinar cells undergo apoptosis by cleaved caspase-3, leading to the overall loss of homeostasis in the organ and dysfunction. And I'll just briefly highlight, we have some of the molecular mechanism here, especially for live-1 and what types of molecules they're making and how they're expanding the pancreatic stellate cells. I'll go through just subsets of that data in the interest of time. Check again. Yeah. What we find is the live-1 macrophages are producing PDGF, particularly PDGF receptor, PDGF-C. So we would use a single cell analysis from both macrophages and fibroblasts to look at their receptor ligand pairings. What we find is that the live-1 tissue resident macrophages are absolutely great producers of PDGF, and the fibroblasts within the pancreas of pancreatitis are absolutely great receivers for PDGF related signaling. They have that. What that means in translation is tissue resident macrophages marked by live-1 have PDGF receptor C, has PDGF-C, and PDGF alpha and beta are expressed in the fibroblasts, and they signal through this, at least transcriptionally, according to the single cell. There are also some other unique aspects of the signaling network. The fibroblasts make a lot of CSF1 in the context of pancreatitis, and perhaps the most responsive to CSF1 predicted by the single cell receptor ligand pairing and signaling is actually the live-1 tissue resident macrophages, which may explain their hyperexpansion during pancreatitis. And then we've done some experiments around this showing that inhibiting PDGF receptor through a RTK inhibitor actually blunts fibroblast expansion and pancreatitis and leads to overall animal death in the context of cereulin-induced pancreatitis in the severe model. So taken together, these data suggest that macrophages of multiple origins exist. Tissue-resident macrophages may have unique tissue remodeling phenotypes, at least in the pancreas. And I'll get back to the model slide in a minute. This is a picture of my son. He's asking me what about cancer, because we also did this in cancer. So in short, in one slide, I'll show you the cancer studies. John also did this in several models of cancer. We like genetically engineered mouse models of cancer because they have the fibrosis that we like and that is important for a human cancer. In the context of one of these models driven by HIT and KRAS and a single p53 loss, he gave 2 doses of CSF1 clonidinate as before, and then gave a recovery period, and then followed by cereulin-induced acceleration of this model. Since a single hit of p53 is not enough to accelerate this model. He finds this after about another 45 days. He gets stark pancreas cancer in this animal model, as is expected. This is florid, low-grade, high-grade pancreas cancer with a lot of stroma involved, a lot of expansion of cancer-associated fibroblasts. When we look at the mice who were now 45 days earlier depleted of tissue-resident macrophages, but then left alone after that, what we find is this animal model seems to stall out. It stalls out at some sort of hyperplastic or cystic-like formation, cannot fully go to the stromal expansion and the high-grade, poorly differentiated pancreas cancer, which is characterized by this model. And there's a failure of the expansion of the stroma in these cystic lesions. This is just quantitating that here. Once they do get to poorly differentiated lesion area, then they much look like the other one, but a significant portion of these tumors actually are stalled out early on during progression. So, okay, model slide, suggesting that there are multiple subsets of macrophages present within these tumors as well as pancreatitis, and they have distinct roles. Live-1 tissue-resident macrophages appear to regulate pancreatic stellate cell or fibroblast expansion particularly in these interacinar areas, and maybe their ability to lay down extracellular matrix in the context of pancreatitis. But the important part of this is that this role is not there solely to promote pancreas cancer. In the context, these tissue-resident macrophages, in the context of pancreatitis, they favor homeostasis and survival. This is their role: help repair the pancreas, help preserve homeostasis, help wall it off with fibrosis if it's chronic. And that preserves the survival of the organism. But in the context of pancreas cancer, this over time will favor pathogenesis in cancer development, as we can see in cancer models, suggesting the same tissue-resident macrophages and their ability to remodel the extracellular matrix and expand fibroblasts has dual roles depending on the exact consequence or context. Okay. What about translational cancer stuff? So now we're going to totally flip into a totally different story. Good. So I'm going back to this slide with the GSE analysis across macrophages, comparing pancreas to all the other cancers we can get our hands on. And what we find is, sorry for the spelling, decreased interferon and ROS responses, at least by GSEA, present within the pancreas cancer tumor-associated macrophages. And we were interested in this. Is this a pancreas-specific problem? And if it's a problem, then this is what we should come at pharmacologically in this disease, or at least test coming at pharmacologically in this disease. Thanks in part to Lisa, we utilize a serial multiplex IHC platform that she's really spearheaded. We don't tend to do 35 markers like she does. We're more on the 12 marker range. This is just going to show you, because I'm going to show you a bunch of images using these cellular phenotypes, but we can co-stain for, say, CD8, C68, or CD163, STING, which will become relevant later, or CK19, and we merge all these images to a composite image. We then use a couple computational methods to assign cellular identity, and with this, you can mark multiple different markers in a cell that has X, Y, and Z parameters, which will become readily apparent and relevant in a minute. We do this in human and mouse, and we use them to assign the cellular identities I'm going to tell you about it. So this is looking at this. We've done this in a couple different ways. So from the GSEA, we would predict that both STING signaling and interferon signaling, as well as STAT1, are predicted to be down or at low levels in human pancreas cancer tumor-associated macrophages. So we look by IHC for a little bit of validation, since the transcriptomics can lie. So we looked at STING positive or STAT1 positive macrophages as marked by a couple markers, but I'm showing you just CD163 for simplicity here. What we find is this is actually true in pancreas cancer. The majority of macrophages present in pancreas cancer, they are either low or negative for STING as one element of interferon, possible interferon induction signaling, as well as STAT1. And this is just showing you how this correlates with the T cell responses within these tumor microenvironments. When you have high STAT1 or STING positive macrophages present within pancreas cancers, they tend to have higher T cell infiltrations in the surrounding areas. And the number of STING positive macrophages or STAT1 positive macrophages tends to correlate with overall better survival in our TMA analysis of human pancreas cancer patients. So this suggests that maybe this is a problem, at least in pancreas cancer, and we could go after this. So I'm going to sidestep for a minute to get at how we're going after this, because that's not what we were thinking maybe 4 or 5 years ago when we started looking at a CD11B agonist. So we first looked at a CD11B agonist, which I'm going to show you activates the interferon signaling in a minute, because we were looking at ways to blunt myeloid responses within the tumor microenvironment. And a very nice novel approach was spearheaded by Vineet Gupta at Rush University, which is in Chicago, so not very far away from St. Louis. And while CD11B is an integrin expressed on multiple myeloid cell subsets and occasionally on adaptive immune cells, and it's actually pretty important for cell adhesion and transendothelial migration, at least according to the papers out there right now. Blunting of CD11B by antagonists had been already tried, and you cannot saturate this target at pharmacologically safe doses, at least according to what was tried 10 years ago. So Vineet developed a molecular agonist, an activator, or an allosteric activator of CD11B. And what he found was this in inflammatory models, but not cancer, could block the number of myeloid cells infiltrating into these inflammatory conditions, such as kidney inflammation and thus preserve some of the homeostatic function of those organs. We were interested in this because it might be able to blunt the number of myeloid cells present within the tumor microenvironment. In our early studies, in our early studies using various different cancer models, predominantly pancreas, but also lung and other models, suggested when we give a CD11B agonist, which at this point is going to be called ADH503, in a minute it gets a new name, and I apologize for that, but it was commercialized. When we look in tumor models and we treat this for 10 to 12 days, we can find that indeed the CD11B agonist can blunt multiple subsets of myeloid cells infiltrating into tumors, including neutrophils, monocytes, eosinophils, and macrophages, or monocytic and granulocytic MDSCs, if you like. And when we do this, at the time, 2019, what we thought was happening, we're limiting the number of myeloid cells in there, and we restore the T cell responses to a much higher number in terms of total CD8 CTLs within the same tumor microenvironment, but also better effector function measured by Ki67, or in data not shown, granzyme or perforin. And they also have increase in PD-1, and they suggest that suggestive data more recently has suggested they're more checkpoint activated. There's also some changes in dendritic cells, CD103 dendritic cells that don't express a lot of CD11B, and they actually increase in number within the tumor microenvironment. So in past thinking, we're blocking myeloid cells. That's what we thought we were doing. We find that we blunt myeloid cell activation, myeloid cell number within the tumor microenvironment, and we can restore partially T cell responses and improve DCs. So then we look for efficacy in these animal models. One, it's good, it's on target efficacy. So CD11B agonist can blunt tumor growth. It doesn't cure these tumors. This is a sub-Q pancreas cancer model, just for simplicity. In the presence of a CD11B knockout mice, we don't have this biologic effect. And before I move forward, I should also say parallel studies in other models with similar sort of findings were also done by Judith Varner and mirror some of our findings as well. Then combine this CD11B agonist with multiple different lines of therapy. Here's combined with PD-1. And what we found is that improvement in T cell led to better PD-1 responsiveness in these animal models. So CD11B has some biologic effect, PD-1 has none, and the combination leads to regression in both of our models, and in one of these curative regression, and the other one they eventually progress. And that, at the time in 2019, was enough to move this forward clinically. So ADH503 became GB1275, and a phase 1 dose escalation with and without PD-1 in refractory cancer patients was opened. And we were lucky to have this trial also here at WashU, and we ended up enrolling quite a number of patients. And at the time, based on Gossamer's approach and our knowledge at the time, the primary biomarkers were things like increase in TILs and a reduction in CD11B positive cells or macrophages. But there was something that really bothered us over the time, was that it took us about 10 to 12 days to change myeloid cells present within all the tumor models we looked at, which is about the time at which we get some turnover of these myeloid cells. And macrophages were on the 12-day range, neutrophils were more on the 8-day range. But if we looked at efficacy, we saw direct efficacy in CD11B dependency within the first 3 to 5 days, suggesting that we are doing something very different. And maybe that's important to the eventual clinical study. Maybe CD11B is having some direct activation phenotype on CD11B positive cells and tumor-associated macrophages. And we were augmented by this by some transcriptomics we had done, both in vitro and in vivo, looking at the effect of the CD11B agonist on macrophage phenotype, where we saw an augmentation of several chemokines indicative of interferon signaling and a downregulation of IL-1 signaling across the board. So enter a really talented postdoc in my lab, Zhu Ting, and she was interested in studying the molecular mechanisms of how the CD11B integrin activator, or allosteric integrin activator, is really changing interferon and IL-1 beta signaling. It's a little bit counterintuitive for how we think about how those things are regulated. And by actually very careful biochemistry and cell signaling that I'm not going to show you, that took her a large portion of time, that I'm only going to show you in a schematic, this is what she's found very robustly. The compound activates allosteric activation of CD11B, and in a CD11B dependent way, activates FACROC signaling that leads to ROS and mitochondrial dysfunction. This ROS mitochondrial dysfunction is not enough to kill the cells, but does release a small amount of mitochondrial DNA into the cytoplasm that pre-activates cGAS-STING signaling, and with other augmentation can lead to full IRF3 translocation, phospho-STAT1 activation, and interferon gene regulation. The pathway for IL-1 is completely distinct. By moieties we don't actually fully understand. It targets NF-kappa B P65 to the proteasome and leads to its degradation. But by a single hit on an integrin, we end up rephenotyping macrophages by these two pathways. Now, and there's some crosstalk between these pathways. So when we hyperactivate STING high enough, we actually get reactivation of NF-kappa B. But I'm not going to talk about that today. But this is important because this would allow us biomarkers for both who we should treat and possibly, sorry, I got a reminder on my screen. We'll see if it goes away. Who we should treat and possibly what we should look for biologic activity of this novel myeloid interactor, because this is not a kinase inhibitor. You can't just look for the downstream targets. So when we looked at this, we first looked at this mouse, and I'll show you the human data. What we find is that we can see this in vitro. Of course, there's nice biochemistry and nice molecular biology shown there in a schematic. But we absolutely can see this in vivo across multiple models. What we find is whether we're marking STING or STAT1 in CD11B positive cells or total macrophages, we find an augmentation of the number of STING positive TAMs present within the tumor microenvironment after treatment with the CD11B agonist. Same true for STAT1, and a downregulation of nuclear NF-kappa B T65 present within the same TAMs in the same tumor microenvironment following treatment with the CD11B agonist. So the mouse model recapitulates what we can see in vitro systems. And then we looked in the context of that phase 1 clinical trial, and we were fortunate enough to get 11 pre-post biopsies back from patients. So all refractory patients treated with either single agent, and a subset of these are actually treated with single agent in combination with PD-1. Subset analysis doesn't matter. Across the board, we see in the majority of these patients that when we look by multiplex IHC, we see an augmentation in the number of STING-positive tumor-associated macrophages or STAT1 positive tumor-associated macrophages in the majority of these patients. And when we see elevation in STING or STAT1 positive macrophages within the tumor microenvironment, particularly these high levels, we see an augmentation in the number of CD8 TCLs present within that same tumor microenvironment, particularly in close proximities. So these data taken together suggest that the cell signaling through activating integrin with the CD11B agonist, we get an augmentation of interferon signaling that we can translate into biomarkers, at least early stage partial biomarkers within the tumor microenvironments of refractory cancer patients. Now, how can we exploit this? Another way to say this is, what are the amplifiers of this biologic effect? So first we had to understand some of the context. And one of the contexts I sort of didn't hint at here is that we only get this if you drop the CD11B agonist on monocytes, human or mouse. You absolutely won't get an induction of interferon response. You'll get a downregulation of NF-kappa B, but not an induction of interferon response. There's a context that matters for this. And the context we find is that the tumor conditioned media, which we use to model the tumor conditions or tumor-associated macrophages directly isolated, are critical for this induction of interferon response. So naive macrophages don't get this response, which may be important for the overall safety of this compound. What we find is when we looked at the tumor conditioned media, our original hypothesis is there's some polarizing cytokines that must be involved or critical in this biologic effect. It turns out it's the metabolites. So to do this, she did some size exclusion columns and just basically looked across interferon genes. And when she looks at the small size metabolites she finds from TCM, they're instrumental in the ability of this CD11B agonist to augment interferon response. And what she found over a course of several studies is the key TCM metabolites are actually DNA, both released these are switched double-stranded DNA from dying cells and the little bit of mitochondrial DNA that also flows out at that time. Now, in most cell culture mixtures, this is like a small percentage of cells that'll die at any given time. But if we treat with chemotherapies or radiation therapies, we can augment this further. So we treated the cancer cells with chemotherapy or radiation therapy or various different killing agents. And then we took that conditioned media and applied it to the tumor-associated macrophages, and then afterwards treated with the CD11B agonist. What we find is this dramatically enhances the ability of the CD11B agonist to then augment overall interferon response. Here's a pretty robust interferon gene induction. And in data I'm not really going to show you, we've done this in a large number of ways. It turns out, so first, CD11B agonist actually turns out to prime STING CGAS signaling. It doesn't fully do it robustly enough to have a large interferon induction, which is probably perfect in the context of not wanting interferon induction across macrophages across the whole of the organism. What we find is whether it just be tumor cell death mediated by chemotherapy or RT-derived dsDNA or mtDNA, or whether we use TLR agonists, or whether we use STING agonists or other moieties, we can then further augment this as though the CLMB agonist is lowering the barrier for which these compounds then can absolutely induce interferon at a very high level. It showed the in vitro data, the in vivo data is as impressive. So this is looking at the chemotherapeutic effect, combining the CDLMB agonists and chemotherapy. This is gemcitabine and paclitaxel, standard for pancreas cancer. They both are effective at restraining tumor growth. The combination is great, and there's a survival benefit. We've shown that before. When we looked at these tissues for overall interferon gene expression, what we find is that both chemotherapy and the agonist can mildly induce interferon expression at the tissue level, but the combination of the two actually gives us sort of like massive fold inductions of interferon genes, primarily through these, in a STING-dependent manner, through these pathways. So what did I put here? What this story suggests is that one, CLMB agonist has multiple effects. There may be some blunting of the trafficking of monocytes and neutrophils, but the primary biologic effect that we see, at least in animal models, and we think possibly in human, maybe through its direct activation. This direct activation primes the pump for STING, CGAS, interferon signaling. Now that alone is not enough to it only marginally helps this, but in the presence of dying tumor cells, which would be present in the tumor microenvironment, or the chemotherapy or RT-induced dying tumor cells, or if we give agonists like TLR 7/8 agonists or STING agonists, we can absolutely overcome the normal threshold of resistance in tumor-associated macrophages and get really robust interferon responses. We think this is exploitable, and we have written to basically open a trial to look at this approach. Okay, these are the two stories. Hopefully I'm not okay on time. Story one was about origin and architecture. So macrophages, tumor tissue-resident macrophages regulate fibrosis induction. They protect from pancreatitis. That's why they're there. That's why they're biologically present in the pancreas. That's what they're doing. But that promotes tumor pancreas cancer pathogenesis over time as a sub-consequence of their biologic protection. Integrins in translation, integrin agonists are a novel approach for reprogramming myeloid cells. 11B can't be the only one. 11B agonists can prime STING interferon signaling, and this can be augmented further by other approaches, and novel combinations are possible, at least we hope. And these are the two stories I thought I'd share today. Of course, I've tried to highlight the people primarily doing the work in the lab, but this is absolutely a collaborative event between multiple WashU investigators, as well as collaborators pharma and in academia outside of WashU. And as Lisa mentioned, I'm totally looking for talented postdocs in the lab. Give me a holler if you are looking and this type of thing interests you. And with that, I will stop sharing.
Tour de force, David, remarkable. Let's open this up for questions. You can put your questions in the chat or turn off your video and or turn on your video and ask David directly. Judy or Dimitri, do you have anything?
Yes, yes, I have a question. Yes, I think this is fantastic presentation, David. Thank you. However, I have some difficult question because we were burned with the STING approach. We know how it works and how it doesn't fulfill expectations. So your idea of sensitization is quite nice and very good. What I noticed though in your mouse model, that effect you presented was very modest anti-tumor effect because tumor basically grow with the same speed. It's just some delay in tumor progression. My question to you is, what the mechanism of CD11B agonist anti-tumor activity you think it happens? And the related question to that is, do you have any model where you have anti-tumor effect which is sustainable?
Yeah.
So basically in mice, delay in 3-4 days or improvement of survival, which you showed is okay, but it's not really impressive. So do you have anything which worked that we can see as the mechanism of action?
Yeah, so couple answers there. First, let's talk about CD11B agonist is not monotherapy. it delays tumorigenesis, tumor progression in our mouse models. That's nothing to write home about, right? What we think about it is a way to reconstitute the tumor microenvironment through the myeloid cells and allow for other therapies to be better. So you're right, single agent delay is not impressive. Sting, let me make sure. I'll try to get to all, you have like three parts of this question. It's a good question. Sting, I'm going to say something and then you're welcome to correct me. You know, Sting agonists had a lot of promise, but there are two deleterious drawbacks to this. Global inflammatory augmentation through Sting. It's not very specific, right? And local versus systemic. Most of them given systemic is toxic. Local only is going to get wherever you stuck it. So these are problems for Sting alone that we think that maybe myeloid biasing here through an integrin agonist in this example might get you around a little bit. We don't see augmentation of Sting in, say, Kupffer cells. So it's given systemically, but we see local activation within the tumor microenvironment, primarily because it needs a second signal. We think that's possibly something we can exploit. We find in combination, we get quite good striking efficacy, dependent on the model. And I say dependent on the model because it requires a context-specific event. You need that second signal. You need the chemotherapy or RT to be killing the cells, or in immune checkpoint combinations. We can cure some models and get minimal effect of the combination in other models. And we think that has to do with how the cells are dying normally without chemotherapy. Our clinical approach to move this forward, if we can get it funded, clearly we're trying, right, is to put a chemo backbone, the myeloid agonist, plus and minus PD-1, and then determine whether, one, we can see the same biology in the context of chemotherapy, and then whether we can augment this further with PD-1. We won't, I think at this point, we can do more mouse modeling, but we're not going to know until we look in human. And with 11 biopsies, that's pretty good data, but that's as much as I've got for the clinical studies that were done as part of the phase 1. Not full answers. Does that at least address your question, if not satisfy? It's a fair point, Dimitri.
David, I had a question with regards to the CD11B agonist studies. You talked about impact on the macrophage specifically. What's it do to the neutrophil? That is surprising to me. What's it do to the neutrophil, or did you look?
So there's what we thought it did and what we in the neutrophil, there are parts of this that are what we thought it did a couple of years ago, and then there are parts of this restudy that we have refined this. When we look at trafficking studies, that is to bead label neutrophils, monocytes, whatever, it will blunt their infiltration in mouse tumor models. When we look for direct biologic activation on the neutrophils, it doesn't seem to do anything. Single cell analysis, tumor microenvironment comes back with 10 genes. So we don't see a biologic impact on the neutrophils that we can measure by gene expression, other than possibly trafficking.
But what about nets? I would almost think that you would impair a post-translational impact like netosis.
So that's possible. We've done another study. That's possible. There are possible things that we don't see with transcription that could be activated. We've done another study looking at the CD11B agonist in the context of the Sting knockout, bone marrow transplant, this context. And this was actually informative because we still see a blunting of monocyte and neutrophil influx into the tumors, but the macrophage reduction absolutely requires Sting, suggesting that plugging in the tissue residency part. But these long-lived macrophages are actually turning over not because of monocytes, but because Sting activation leads to their decreased longevity. But the fact we didn't see that problem in the neutrophils suggests that at least Sting isn't involved in a pathway that leads to their reduction in number. So there's a numerical thing that happens over time, and then there's some on-site signaling that happens within macrophages. We haven't been able to assign that to any other cell type.
Okay. I think Ho-Quing Ying has turned on his camera. Do you have a question for David?
Yes, thank you, Lisa. That's a great talk, David. So I have a question about the first part. So you showed the live one has a very distinct spatial distribution. Does it also cause a spatial distribution of the fibroblast? And the second question is the deletion of the I think that you showed the depletion of those level 1 positive cells to early death of those animals following cellular injections. I'm just curious, what cells died off because of the pancreas cannot regenerate, or it's because of the epithelial, like ATM lesions or other cell death?
Yeah, so I'll answer the second one first. When we do a little bit of autopsy and we study their blood circulating levels, it looks like severe pancreatitis as the cause of death. And then we've done studies in the past where we did surgical resection of 85% of the pancreas, try to create a metastatic model. And the cadence of this is identical to loss of pancreas function. There's no way we can eliminate all the other possibilities, but we don't see severe problems in other organs, especially at time points prior to death. When the mice die, they have multiple organ failure. So simply losing the pancreas is enough to kill the mice, and we see liquefaction of the pancreas with the combined TRM depletion and the severe pancreatitis model using the pump. Your first question was about fibroblasts, and the live ones are in that interacinar area, and that area is where a large number the live one macrophages, or a large number of fibroblasts are present. But I didn't understand the rest of the
like whether, because as usual, there's also a specific crosstalk between live one positive macrophage and I think the ICAS, right? So I'm just wondering whether the live one macrophage also determine there's a spatial distribution of certain fibroblast subsets.
That's an excellent question. I don't think because we numerically see less of them, I don't think we've looked at their spatial distribution in terms of the fibroblasts after TRM depletion or live one TRM depletion, but they may change. There's a little less space in the mouse than there is in the human. It's a shorter contexture of the fibrosis in that model, but that's a great question. We should look at that, and in cancer, because we've done it in cancer models too. Thank you for that.
Thank you.
I have a question. Both your stories were just beautiful, so thank you for presenting those. I had a question about your CD11B work. The mechanism you described is really fascinating. Do you think that's an aberrant response, or do you think that that is a natural pathway? So can you reproduce that, for example, by clustering CD11B integrins with antibodies, or by allowing them to adhere in macrophages that are appropriately conditioned, or do you think that's an aberrant response due to the effect of that particular agonist on the?
I think it's
molecule?
a aberrant response.
Mhm.
That isn't seen. So when we do this with ICAM or something like that and try to cluster with extracellular matrix, we can't see the augmentation of the interferon part as well. And you may have done this too. You've done some of these studies as well.
Yeah.
So it looks to be a molecule and conformation specific event that we ironically got in this molecule. I know there are possibly other molecules that can reproduce this now. And I think that suggests it's not a unique biologic function to this structure, but if you can get that integrin in that structure, you can get this biologic effect. I'm not sure that this happens naturally, or if this happens naturally, it's sort of a much more subtle thing that can't be augmented to this degree. We can't reproduce it by clustering, and even our antibody clustering doesn't seem to do that either. We don't get the opposite effect with a knockout either. So this isn't a,
So I just had one other question, whether you see any association of CD11B and a TLR, because it had years ago it was described as it being a co-receptor for TLR force, but I wondered whether it could be associated with one of the other TLRs to produce this outcome.
there are two possibilities we think are. There are two possibilities that might involve TLRs. We are now testing the changes of the TLRs that we find that it can augment quite greatly, that maybe it upregulates the TLRs or their surface availability, or even their availability. Anyway, that's one possibility. The other possibility is that clustering is different. That could happen two ways: because of CD11B's activation, or CD11B, we're activating alpha M and we're squelching some of the beta units to do that. and maybe we're changing the integrins over here.
Yeah.
I've never been able to explore that, but I think that's totally possible because of the amount of bringing in that we're doing, the amount of adhesion we're generating or activation. I don't know the answer, sorry.
David, there's a question in the chat asking if you've tried any combinations with CAR T-cell therapy with your 11B agonist, and also if you've tried to combine with any other immune modulating agents thus far, checkpoint inhibitors, vaccines, etc.
Perfect question. We haven't done CAR T cells. It's a warranted question, but that's not our specialty. We, as sort of hinted at, we have put this with STING agonists as well as TLR agonists to just proof of principle, see that those agents can be improved. In the responsive models that we published in 2019, the most hyper-responsive models, we did CTLA-4, PD-1, and 4-1BB. Does not work with CTLA-4. That actually kind of makes sense in retrospect. It did work with 4-1BB, and it did work with PD-1 in terms of augmenting their efficacy. And that's what we've tried to date. Last few years, we focused on the mechanism. That's what postdocs worked on, because we thought that might be important if we're going to move this forward. We're going to try to move this forward. That may
Right.
be.
Thank you. There's a hand up by S. Shetty. Do you want to turn your camera on and ask a question?
Can you hear me? Can you hear me?
Yep, go ahead.
Oh, hi. Thank you, David, for a fantastic talk. I just wanted to go back. I'm sorry if I missed this. The live one positive macrophages. Do you think that process is driven by hyaluronan? And is pancreatic stroma known for high hyaluronan expression?
So yeah, so pancreas cancer has a ton of hyaluronic acid. I'm not sure that expansion or their number, I don't know. We haven't looked at HA levels in the pancreatitis model. I think that possibly their retention and their expansion has something to do, and their localization has something to do with the extracellular matrix, as in they have certain adhesions which would keep them there. Why not move over to the acinar space? There's got to be a reason. It's either that they love some cytokine the fibroblasts are making, which is they make a lot of CSF1, but that's not a really good reason, or that they really like the adhesion there. But I don't know whether it's HA. I had a snap answer that I'm not sure anymore, but it's totally a ton of that in pancreas cancer, as the fibrosis is much more expanded, and in human chronic pancreatitis. In the mouse model, our chronic pancreatitis model gives us some stroma, but not as much as somebody who's had it for 10 years.
Thank you so much.
So I can't answer your question the right way.
Sorry, thank you.
Okay, I don't see any other hands up or any other additional questions in the chat. So thank you, David, so much. That was spectacular, quite a tour de force. Please extend congratulations to everyone in your lab, and we look forward to seeing you soon.
Thank you for having me.
Yeah. Hey, Judy, can you stay on for a minute?
Yes.
Great. So these are all the people that walked away from their computers.